And the fish has retreated.
A lot of little hiding spots in there.
Yeah, wow. Okay.
Yeah.
We'll have to pass on the fish.
Okay.
But let's check out our corals here. Again, that's a sponge on the left, but the one on the right, well, actually the one on the right also looks like it might be one of these sponges. Let's zoom on one of them. Maybe this is just easiest here.
Okay, we'll zoom in, please.
So, this is a sponge?
Yeah.
Oh, what?
Can we try to sample a piece of this?
Yep. Yep, I think we're all the ship's stable height.
Okay.
Okay.
I think
15-ish, yeah.
Maybe a snip and slurp.
Yeah, do a little snip. Okay, we'll come wide, please. That would be a good time to do a Niskin if you wanted as well.
Oh yeah, go ahead whenever you can.
Has anyone sampled a brachiopod from this region?
I have no idea.
If I grab the arm?
I think Max wants to sample one.
Are we going to do coral sample first, then Niskin?
Whichever order you guys want to do it.
It doesn't matter to me.
Do you want to quickly do the Niskin
Doesn't
order?
matter to me
Sure.
either.
Let's do the Niskin first, yeah.
Okay. Just give me 1 sec here.
I gotta come up.
Yeah, can we, I'm going to go down anyway, sorry. Can we just back the ship up 5 meters? So, sorry, that would be, which way did we go before? Towards the wall?
I have not changed my heading, so.
Okay, so reciprocal, so like 7.5, I guess. Reciprocal of Atalanta heading.
Which will be like, yeah.
Like, Atalanta heading's 250. Yeah, just 5 meters.
Oreo.
It's an Oreo, yep. They are cool fish.
Okay. I'm gonna start moving over there.
I like how they swim.
Yeah, I really do too.
Are you retracted?
I am.
Okay, perfect.
Which bottle number are we tripping here?
We're going for
We need
2.
to trigger 2?
Number
If
2,
possible,
okay.
yes.
Yeah. Is everybody watching?
We're all watching.
Is there a reason why some are balls and some
Yeah.
are
Just
wicks?
triggered.
Thank you.
826.
Okay, I gotta come up.
Okay. Yeah.
And right there is the colony.
Yeah, okay.
Looked like there was one lower down too, but there's a few of them.
Right, we're gonna do a snip and slurp, so I gotta.
So, yeah.
Yeah.
Okay, I'm gonna perch out.
Okay.
That's all I got. Okay, so that was the one straight ahead?
Yep.
Okay. Turn it down again. Like if you turn it up and you do something, well, I just gotta remember to put it back.
Okay, sounds good.
Yeah.
Oh yeah, because it messes
This
with
is
all
the
your
camera.
camera
Yeah.
stuff.
Maybe if you do this, maybe kind of a bit on an angle.
Max, did you preserve that brachiopod we caught 2 dives ago?
No, I was unaware that we caught a brachiopod.
Oh, I thought we showed that to you. It came up in the rubble.
Is it still around somewhere?
I presume so.
Oh, man. How thrilling.
If you ever came and visit us in the lab, maybe you would have seen it.
That was spicy.
Spicy.
Just me encouraging Max to come hang out more.
Oh my goodness.
Well,
Are we just gonna do a flying?
we might have to do it
snip?
on the fly.
That's
Yeah.
fine.
Okay, let me fix my camera.
Make sure the arm doesn't.
Okay. Which part, because you see that there's that kind of dead part on the end. Do you
Yeah,
want,
no, I want more of the live part.
so more of a white piece?
Yeah, I can't quite see the ends here. I'm trying to find something that's out on the end, but if you drop down just a touch.
Let me pull back the porch, actually.
Yeah, I mean something there
Okay.
like that
Yeah,
would be
that's
just
doable.
fine.
Yeah, like one of the branches or whatever.
Yeah. Maybe 4 or 5 inches of it, something like that.
Yeah, okay.
Okay. You want
That's
to
a
try
pretty
that?
good spot
Yep.
for me. Yeah.
What did you think this one was, Jeff?
Are you ready if I
Yep, ready.
Hang on a second, and I will give you a name.
gonna come in here? I can see I need to be more out here. I go for this one. Okay. Which is,
Massive.
It could be Walteria. There's
So don't go too long of a piece.
yeah, you want
It's
like more
a
like
different
that?
kind than
Yeah,
the
even
more
shorter
tubular
maybe.
ones,
Okay.
but
Gotta
Walteria.
fit in the jar.
Maybe I'll go for
I think we have
this
focus
piece in here.
issues again with the camera.
Sure.
see if I can line that up.
I haven't managed all day to take
To get it
a
focused
perfect
well.
sharp one.
Yeah.
Sorry, gotta get over the other side of it. Oh. Here, how's that?
Sure.
Might have got a little bit more of one of them.
You've got a piece drifting there.
Yeah, I see that. Here, I'm gonna take
Right there.
this one.
Yep.
Yeah, there
you
we go.
got it.
Okay, I gotta back up a little bit. Okay, I'll porch out.
Can we put that up for a close-up before we
Yeah.
suck it up?
Fix that.
Just gonna go slow so it doesn't break off.
Yeah, I'm just gonna back up just a little bit first.
Yep.
Looks like
Jaw
we've
lock.
drifted even further away. Yeah, jaw lock for sure. Let's do a 5 meter move on Atalanta heading, so 250, 5 meters. Okay, yeah, we can go for camera shot of it.
Yeah, I go for zoom video. Let's get in close. Let's take a look.
There we go. Excellent. Thank you.
Okay,
Yeah.
wide. you want me to porch in a little bit? Or are you happy there?
I think I'm happy here, actually. I can get my pitch around the other way.
Okay, let me give you a shot of it here.
Yeah, and I can
Just gonna
see.
get it close to the tube. Down.
Yeah, you want to pull suction?
Yep.
on?
Yep, ready for that.
Yeah, that way it just
We're on 3.
will probably.
It's on low.
Yeah, alright.
You can zoom.
Can you
zoom.
go for a half zoom video? Just trying to tell if it's actually in there, if it's...
It is not.
No?
Oh, no, yes. I'm not sure.
Oh,
I got confused.
yeah,
I thought
I
the
know.
sediment there was...
I know.
It's on
Can
50.
we bump zoom suction up like
Yep.
a lot more?
Yeah, it'll happen. It'll go.
Yeah.
You're just kind of far away from it, I think, right now. Getting there. There we go.
There we go. I think that's in there.
Yep.
I'm gonna get it all the way to the end there.
Hold on a sec.
Yeah.
Good?
Yeah.
Okay, ready?
Yep.
There we go.
Nice.
I gotta slow it down just a little bit so we don't.
There you go.
Okay, stopping it.
Thank you.
Yep. Okay, and then I'll jar rotate.
Coming up.
Okay. Oh, I'll go down. Sorry, I was drifting up a little bit.
Okay. Okay, putting the arm away.
Okay.
Okay. Nice. Okay.
1004.
So
Oh.
No, what is it?
yeah, Tinophore. Oh, wow. Can we zoom on that?
Yeah.
I'm coming down to give you more.
Okay. We'll
Good
zoom
zoom.
in, please.
Oh, wow.
Yeah, this is great. This is a comb jelly. Looks like a baroid.
Whoa. That's really cool.
Why does it have that long sort of trailing thing?
Yeah, so it can use those for prey capture, apparently. But baroids mostly just engulf their prey with one end is just a big opening. And
What do they eat?
other
Okay, come on,
jellies,
please.
actually.
Oh, no.
Yeah.
This is
Oh,
on
that's fantastic.
that was on my wish list.
There it goes.
Cool.
Well, let's keep proceeding along the wall or up or down the wall, whichever best suits the ship and the vehicle, Josh.
Okay. Let's do just a 5-meter step again on the 250
Look at
again.
the size of that colony down there. Wow, down and to the left, there's bunch of stuff.
Have we seen this level of brachiopod saturation on other dives?
No, this is the first.
Max,
And-
are they filter feeders?
Yes,
Like
they are.
bivalves?
Exactly
everything?
like bivalves, except for nothing like bivalves.
Well, they've got these.
We'll zoom in, please.
It's like the abstract pie all over again.
Yeah, they're an abstract bivalve. Or really, bivalve's an abstract brachiopod.
Great. I understand
Nice
completely now.
bamboo.
Brachiopods have been doing it for longer, I believe.
If we can get more of a zoom on the end of the polyps, that'd
Sure.
be great.
Are
A
these
lot of it's taken over by zoanthids or something there.
so sort of a parasite? Use
Yeah.
the structure of the coral.
Oh.
But Jeff, you were saying we don't know if the zoanthids, like, kill the coral or not.
Yeah, they may just be taking advantage of open space that
Yeah.
was, you know, made free. Maybe a sea star came along and ate part of the bamboo coral and it's just taking up part of the space. Yeah. Not entirely clear.
What is that?
That's Oreo in the background. If we pan a little to the right, if we can see, look at where the nodes branch, that's the other part that the taxonomist can use to identify who we've got here. So yeah, okay. So it's branching at the nodes itself. Okay. Okay, let's look at the
come
big
what?
colony to the left.
You might need to come down a tiny bit, maybe.
Ooh.
Let me know if you want a heading change to look at you, but otherwise I'm going to keep it.
That's fine.
Yeah.
Yeah, let's zoom in, please.
Wow, this is quite the bamboo coral. And it is just covered in other animals growing on it. We've got clusters of zoanthids, crabs, some brittle stars. There's some sort of corallomorph anemone on the right, which is actually on the wall. Looks like some little hydroids. Wow. Primnoids growing on top of the bamboo. This coral has been here for a very, very long time.
Can you give us an estimate?
For bamboos, no, I can't for sure. It's hundreds of years, but I can't be more specific than that. Lots of anemones.
What, the big red one there
Yeah,
from the
with the
branches
white
and
tips
anemone.
on there. Yeah. Oh, right over there. Yeah, that looks like a closed up, behind the sponge, that looks like a closed up Venus flytrap anemone. There is so much growing on this bamboo. And out at the end, it's still alive.
Oh, the fish.
Oh, can we, that's an epigonid. Okay.
Another
We
cardinal.
saw that one in midwater, and yep, that's another one right there.
Okay, we're just
Okay.
under 15 meters.
Okay, well, come wide, please.
Crinoids on the
Okay, I'm
end.
going to come up a little bit.
Yeah.
Okay.
Is that a tunicate?
Yeah, not sure. If we can we zoom on that, Josh? Maybe not.
Yeah, I'm just going to
Okay.
go, we'll
Let's go,
keep
let's
on.
go.
Yeah.
Yep.
You do what you gotta do.
I don't want to get in that
Yep.
same spot
Yep,
again.
yep.
Yeah, we're coming up. Okay, I'm coming up faster.
Okay. Is the ship stable?
So many
Look at all the
caves.
Oreos. Couple of More of this
Whoa.
Walteria sponge. So unusual seeing us, for me, to see a sponge that is looks like a
try and
coral.
slide over towards
Yeah,
you.
I've been.
I don't understand the life history of this.
Yeah, you're probably going to want to come
Yeah.
closer.
in,
'Cause
in school, they taught me that you need to have
See what-
osculus
Instead of an
sponge
overhang, I
to
think.
distinguish
Yeah.
it
Right.
from other things. And
I
where
know.
are the osculus in this?
Can we back the
there's
ship
no
up 5
holes,
meters,
so
like
that's why I
reciprocal to Atalanta again?
I think I've got the top in view.
Okay.
I'd like,
Yep.
it's, I think I'm above.
The thing that throws me with sponges too is that, like, that's in the genus Walteria, but the kind of tubular open mesh thing with all the spikes sticking out the sides, that's also in the genus Walteria.
Uh-huh.
And you're right, it's the spicules. You have to look at the
just gonna
structure
pan
of
up
the
to look at
glass,
the-
like that
Sure.
on the
I just
top
want to give us a
left is
bit more time
Walteria.
on the wall here.
And also, it needs to have some sort of hole structures, right, for the filtering.
Right,
You see holes
right.
all over all the sponges. That
Right.
one,
Okay.
where are the holes? So
Yeah.
So we'll
what
step
is
back a little
everything
bit, and
that
then I can
makes
kind of
it
hang
a
out a little bit
sponge?
more here.
Right.
Should be, yeah, there I am.
Some Westler astrid.
Yeah,
holes.
'cause we're gonna
Oh,
wanna look at
actually,
some
if we
stuff
can
here.
look at that right there.
Okay. I think we're good. We'll step the ship back like we just did. We'll wait for it to swing and settle out, and then we can hang out here a bit more. Okay, we'll zoom in, please.
Oh, look at that sea urchin.
Oh yeah. Oh, okay. That's the red thing on the right is a sessile sea cucumber, a solid sea
Oh.
cucumber. They have those in shallow water as well.
Yeah, they're in Puget Sound.
Yeah. I am not sure what the coral is.
Like some
Can
interesting.
you zoom in? Is this a bamboo?
Just give me 1 sec here. Try and get a little closer.
Might have to adjust our iris a little bit too. It's very bright. It's a bamboo.
And you can just tell that by the segmentation? Or?
Yeah.
Yeah.
So I'm going to see if I can figure out which one
Okay,
this
we'll
might
come
be.
wide, please.
Do we want to look at that sea cucumber too?
Yeah,
And the sea urchin.
both.
Oh, there's a gastropod up there?
Yeah.
I'm
Or
still
maybe it's
above
just
the
another
lip, so you're
brachiopod.
Good.
Okay.
So we've got that guy right there.
Okay.
Like a cutthroat eel, bottom right.
Zoom in, please.
Can you try to grab just a screen grab of?
I already
Yeah,
got
I know
a
it may
couple,
not. Okay.
but I can get more.
Maybe Histosideris or something like that, in a genus like that. Great.
Okay, we'll come
Nice.
wide, please.
And if we can look at the red solid sea cucumber.
Okay.
I think its
Okay,
blooms
we'll
are now
zoom in,
retracted.
please.
Yeah. We scared the sea cucumber.
Yeah. Okay.
Okay, we'll come wide.
And
Yeah.
these ones, the one that eat with their,
Their filter.
yeah, they just
Mm-hmm.
put their arms, their tentacles in their mouth.
Yeah, and just wave.
Slurp
Yep.
them away.
Yep.
Yeah.
Oh.
Sea spider?
Yeah. Can we take a look at the sea spider?
Sure.
It looks like.
Oh yeah, that was a dream to see one of these.
I
Hopefully
generally
it is.
try to avoid these in my dreams, actually.
Why do you want to avoid them?
I always found this super fascinating. Pycnogonida, right, is the
Yep.
the name of the...
It's like Megan said, you don't dream of seeing them, but you see them in your dreams.
Oh.
In my dreams,
Can
I
we
see
zoom in,
much
please?
worse things, guys.
Oh.
You know, right?
Oh, wow.
Now, do you know why we're so far off right now on Atalanta? On
These
the
are like,
nav
they can
screen?
be both red and yellow.
Is it just
Yeah.
how it is right
Yeah.
Yep.
And are two
And,
different species?
Oh, there's a bunch. I think the genus is Colosendus, and there's a number of species within it.
Yep.
I'm not certain that their taxonomy is that
Probably-
well resolved.
Probably
Uh-huh.
just a multipath against a cliff
These
wall,
are-
I would bet.
They-
If I'm not wrong, they also live in shallow water, but much smaller.
Yes,
Yeah.
usually much, much smaller, maybe only a centimeter. But in the deep sea and in the Antarctic in shallow water, they get to these very, very large sizes, well in excess of 10 centimeters across. And they
come wide,
typically
please.
use that proboscis out in the front. And they eat anemones, corals, things of that nature. Their bodies are so skinny that their gonads are actually located in their legs,
Oh.
because there's almost no room in their main central body for all of their organs. So they've pushed some of them out into the legs.
Is it
Wow.
in all of their legs or 2 legs?
I'm not sure.
That's interesting.
Even the legs don't
Interesting.
look that thick.
No.
Yeah.
Yeah, let's look at these bamboos here. So many brachiopods on the wall.
The dream dive
Sponges.
before Max.
Okay, we'll zoom in, please.
Yeah, maybe a keratoisis or something like that. But these are bamboos.
Tube
There's a
or what?
tube-dwelling Cereanthid anemone at the top.
Oh, is that Cereantid? Okay.
Mm-hmm. Let's take a look at the one down below.
Okay. Just to come out a little bit.
So many of these Acanthoprimnoa that we collected earlier.
Seems
Wow.
to
Can
be
we
a lot
zoom?
of this bamboo coral. Is that something I would be interested in getting a sample?
Maybe,
You can
maybe.
push
I
in
think
more if you
so.
want.
This is at least the third colony,
Yeah.
so I think we should probably take a piece of it. I think that's a good suggestion, Noel.
Similar kind of method as we did previous one, maybe.
Yeah, please.
In the jar.
Uh-huh.
Okay. Okay, we'll come wide. We'll have to do the same kind of on the fly, I think.
Okay, sounds good.
There's another colony to the right as well.
Noel,
There's a
do you
lot
know
of them.
which sample jars are open?
I do.
What's
One
the lowest
second.
one that's open?
We'll be on 4.
Okay,
For the
perfect.
slurp.
Thank you. Okay, I'm turning up
Okay,
the PRV, but I'll turn it back down.
Roger.
Oh, yeah.
Oh.
Large fish on the right.
Nice fish.
Okay,
Eel of some
4,
kind.
sample jar 4, right?
Yes.
Okay.
I'm gonna retract HD. Down. Heading.
Can we iris up a bit, video?
Absolutely.
Thank you.
Should should we also get the brachiopod by the time we all have all our arms out?
Yeah.
Or don't you think it's very common?
It
we
is
can
very
definitely
common.
say that.
Yeah.
Definitely a representative of the community.
Yep.
I for one
Right.
think we should sample one.
Yeah. Let's try to sample a brachiopod as well, and I see no reason why it couldn't go in the same box with a bamboo, same chamber.
So hold on a sec. We're gonna
Oh yeah, we're gonna snip, right?
right?
Okay.
Yeah.
Yeah. There we go.
Unless you want to do the brachiopod first, then it would be the slurp.
Yeah. What?
Why don't we do that? That way we may not break up the coral, because we're gonna have to continue to run the jar, right?
Okay.
Oh, good point.
Yeah.
So yeah, why don't you grab the slurp,
'Kay.
and then we'll have to do that on the fly as well, I think.
I mean, once you suction a fish on the fly, I feel like
It's
everything
nice
else is.
Something that's stationary.
Yeah. Oh, sorry. Grabbing the. Come on. Go lower. There we go.
Good.
All right. Remind me what a brachiopod is.
I think Max has got this one covered.
I'll
for you.
direct you.
Yeah. All right.
They look like clams.
And
Come
we're
up
on the
just
right
a
jar.
little bit. Are you going to be going down to get it, or?
There's a
Yeah.
whole bunch on the wall.
Yeah.
Yeah, they're
okay.
kind of everywhere.
yeah,
Like, I think
they're
if you
all
just do a
over.
scrape on the wall, you'll get some.
Okay.
What do you want me to do?
Sounds great.
Do you want me to tilt down? Whoa, it's hot.
I definitely turned that down, so sorry, I don't know why.
Well, I turned it up because I need it up for the suction. Yeah, conflicting.
Yeah, let's just get close. Is the arm in a good spot for you to get close?
Yeah, it's just in front, yeah.
Okay.
I am going to tilt up just a little bit. It's going to be a bit of a bump.
Yeah,
And then
it's all good.
So, yeah, it's just the little like dots all over the
Okay.
wall.
The
I might
little
try and
puff.
get one that's on the bit that's poking out.
Yeah.
Jeff, I finally get to use the other classification for our sample types.
Because we don't have brachiopod.
don't have brachiopod. How exciting.
Let's see here. Well,
Are you looking for targets or just maneuvering?
I'm looking, there's so many, but I'm just trying to
There,
find
there,
a good
there.
spot to kind of
Yeah.
reach in. I might actually come up. I'm going to come up a little
Yeah, that way we're not at risk of that coral getting.
bit. And
There's the big coral up there.
kind of want to
Oh,
find
another sea spider.
kind of want to find a
yeah.
spot I can like rest against
Oh, that's the same one.
Yeah.
wall, but it's not
Oh, yeah,
really
it's on
that
the
great of
coral.
spots.
Too good to be true.
See the one where the laser is? See the lasers right now?
Yeah.
Right there in the right laser, just
Yeah.
that one.
Yeah.
Let's go for that. Might just want to do like a partial zoom.
Yeah, okay. I'm gonna pitch up.
Yep.
That way we go in like that.
You want me to
Yeah,
go for
just
zoom?
push in a little more, yeah.
Yeah.
I'll try and get you, I'll try and like basically fly it in there. I'm going to turn on the suction pump.
Okay. And the only thing we're gonna watch is that coral kind of near there.
Yeah.
And you see an upper, there's kind of a rock.
Yep, I see it.
Let me know if you want me to reach
Yeah,
in
I want
there.
you to reach down. Yeah,
Okay.
I think you're a bit low.
And I get-
Yeah, there you go.
There we go.
Yeah.
Still
I
a
can
little
get
ways
in
away.
there.
There you got it.
Hey.
sorry.
Yep, you're close. Yeah, there you go.
There.
Oh, it's bigger than I thought.
It is bigger.
There you go. Nice.
Eh.
It's probably not going to be whole.
Yeah, do we want to get a smaller one?
Yeah.
Okay.
Yeah, I guess I'll keep that running. I'm going to back up this a little bit. That was bigger
take it?
than I thought
That
it
was
was
quite
going
a
to
bit
be.
bigger.
It's quite brittle.
Exciting.
Do you want video to go wide so you can see better?
No, I think this is alright. I'm going to come up so I don't shove the camera into
West
the wall.
Lastard sponge.
Kind of need to focus on the wall a little better though. We're not focused.
Video, can we focus on the wall a bit more? Yeah, that seems smart.
Of course, there's none right there, which would be a great spot.
I like the overhangs there.
There's that, maybe that little,
That little yellow one, right?
maybe that's kind of
Is
under
that
that
one
thing
right there?
I
Yeah.
don't
Be able
know.
to get
Is
it,
it?
It might be a sponge.
Yeah.
Oh yeah, okay. Of course, this is like so much easier, but I'm gonna have to go under the underhang, or overhang. Under the overhang.
Under the over.
Yeah, there's a few back in a cave over there. There's one right here, though. I don't know if that's accessible or not.
Maybe just
not
the
with
ones above
coral.
it, maybe.
These guys here.
Yeah.
Yeah.
Are you going for the one that's
Yeah.
right in the center of the screen?
Yeah,
Or-
right, yeah.
Okay. You might have to go lower.
Okay.
Maybe I can get it. Whoop.
Okay, I'm gonna come down a little bit for you.
Okay. And we are pretty stretched out on the...
Yeah, okay, I'll slide
Okay.
over. I can
Is suction on?
Yep, it's on.
That one's also
Okay,
quite big.
Yeah, I don't know, go for it, you're there.
Yeah.
Even if you just get a piece of it. I don't know that we're gonna get a whole one. They're too fragile.
That one looked empty also.
Okay, I gotta back up.
Okay.
Yeah, we're just kind of getting pieces. Okay, I'm gonna turn that off for now.
Yeah, I think that piece
Okay,
is
let's come
kinda
wide,
stuck
please.
in the-
I mean, we're getting pieces of it, but
Yeah.
Josh, I think maybe we will be done, and let's get a piece of the yellow bamboo now.
okay.
Yeah.
Okay, we'll put that toy away for now.
Yep.
Down.
Can you tilt down
Yep.
just yeah.
Woo, hot.
Thank you. Yeah, we got the spicy camera right now.
Yeah, spicy cam.
Alright.
So I'm gonna go slide back over this way. Are we? Oh, we're at Washington. Okay. Well, we'll hand this
Are
one
we
over
gonna-
to Jake and Sarah. I'm gonna turn this down
Okay.
till the.
They're where? In the what? Oh yes.
So I just get like a piece, like a end piece that'll fit, you know, not super big, maybe just like a. Drawing a part zoom.
Did you say part zoom?
Yeah, we're gonna
Okay.
try and collect this like little end of the coral here.
Yep.
I think that was good there, I think.
Okay.
Yeah, I think it's good. Okay, we'll come wide, please.
Yep.
And then I'll just gonna back up a little bit. I'll show you the. I can porch out a little bit if you want to, unless you're happy there. Can we zoom in, please?
Yep.
That's good. Turn that up. I'm just gonna bring that down. Suction pump. Okay, suction's on. There, yeah.
There you go.
That's probably good.
Suction.
Yep.
So was that's with the partial brachiopod. Is that
All right.
Yeah. Okay, suction's
Stopping.
off. Okay, we'll come wide, please.
Yep.
Rotate.
All right. Rotating.
Oh yeah, perfect.
There you
Okay,
go.
cool.
What was that sample number?
That's sample number 55.
Okay.
And just to confirm, that was with the partial brachiopod?
That's correct, yeah, in jar 4.
Okay.
Actually, we should just double check jar. Yeah, jar 4, I believe, yeah.
Jar 4,
Jar 4,
okay.
yeah.
We had jar 3. Okay.
Okay.
Just so you know, Jake, the intervals are 10 meters on Atalanta right now. The intervals for the sonar is at 10 meters instead of 20, what it usually is.
Oh, okay.
Yeah, just so
Good to know.
so you're aware. Yeah.
Alrighty.
Cool.
Oh, what is this?
Oh, that's a
wow.
Pelagothuria. Can anybody guess what thing this is?
Push in there.
Type of jelly.
Nope.
Squid?
Tinophore?
Nope. It's a cucumber.
Cucumber.
Oh.
Wow.
It's really tricky. It doesn't look like it
No.
doesn't look like a cucumber. But this is a pelagic sea cucumber. It's been specially modified to live fully in the water column. So it kind of looks a little bit like an octopus,
Can we get
a little bit
lasers
like
off?
a jelly.
Please.
But it's actually a cucumber. You can see its mouth parts right there in the center.
Wow.
Wow.
Enough.
What a way to start the shift. Welcome to the 12-4.
The first time I saw one of these, I was like, what is that? As it rotates around, you'll be able to see the little feeding arms. Oh, it's not cooperating.
Yeah, it looks like there's a little something
Yeah.
entangled.
So this is related to the headless chicken monster.
It is.
Uh,
All right, come on.
Yep.
But it looks a little bit more like a vampire squid. You know? All right, so what's the plan?
Yeah, thanks, Megan. I think, yeah, we'll keep moving up here.
Daniel, is that you?
Yeah,
It's Daniel.
You're back?
He's back.
so yeah, excited to be here back on the 12 to 4 after taking a few days off, and what a dive to come back.
Little fish there.
Oh, yeah.
Yeah, if we can try to get a zoom on that guy.
Yeah, you can push in there.
Little rat tail fish.
Little rat tail.
This could be Malacocephalus. Looking at that dusky fin.
All right, come
All right.
on.
Yep,
Yeah.
thanks.
Yeah, that's great. Yeah, Megan, if you can start us off just range to waypoint 3,
Oreo.
just so we get situated.
All right. Our range to waypoint 3 is about 430 meters.
Roger that.
So Oreo is that fish that we were seeing in the last voyage a little more frequently?
Yeah, the Oreos were the ones that were right on that vertical cliff face.
Okay.
That had the sort of wavy fin and or more triangular shaped body.
Mm.
Yeah, the one we just saw, the big eye. Yeah.
Yeah, so just as we get situated here in the new launch, for those who have joined us recently, welcome here. This is the first dive in the southwestern portion of the Palawan Islands, so we've been focusing the first couple weeks working around the northern and eastern and western portion of the sanctuary, and then a couple of days ago decided to come to the southern portion. It was a lengthy one-day trip, and this is the portion of the archipelago that we're going to spend the rest of the expedition on. This is the most remote part of the sanctuary, by far the least explored. It's also based
I
on
think so.
Okay.
the available information quite distinct. This broader region, actually about a decade or so, was described as an ecologically and biologically significant marine area by the Convention of Biological Diversity, just using global databases such as the Ocean Biodiversity Information System, found that the islands are quite distinctive and quite abundant, but that was just really looking at
Push in there.
terrestrial and shallow water fauna.
Oh, it's a tiny eel. It could be a juvenile snapper branchid or a congrid eel.
Nice.
It's hard to tell from this angle. I'm leaning towards congruent because of the depth.
Come on.
Yep.
Sorry.
Yeah, right here up at the top, I bet we're going to have a lot of fish.
The fish.
Lots
Another
of
cuss
fish.
gill.
That one, I think, might be a macurrid. Let's zoom in and find out.
Wanna push in?
Oh, no, you're right, Jake. It is a cuss gill.
Whoa,
Yeah,
Jake.
you got it.
Nailed it.
Jake knows the cuss gills. I'm going to send you a reset.
Yep. Go for it.
All right. Now that
Zoom
we're
out?
not on a cliff.
Do you want me to zoom out, Jake?
Yeah, you can come. You can come on. Okay.
A little shrimp. Maybe.
Yeah, it's a little opossum shrimp, mysid.
Yeah. Cool. Thank you, Daniel, for kind of giving us that little intro. It's taking my thunder. But yeah, we are the 12 to 4 watch, and we're going to start by introducing ourselves. So I'll start. My name is Blodaki Chocho. I am the Science Communication Fellow. And over here, yeah, maybe we can just turn to you and work our way down these chairs, and then we'll get the front.
Yeah, thanks, Blodak. Daniel Wagner is sitting here in the back row, along with...
My name is Elsa Tole. I'm the assisting scientist on the 12 to 4 watch. And then to my right is...
Hello, everyone. My name is May Lubetkin, and I am data logging on this watch and the science manager on the ship. And in front of me...
Oh, great. There was one more back there. In the front row, this is Megan Putz. I'm sitting in the nav seat.
And to the left of Megan is Jake Bonnie. I am the Herc pilot on the 12 to 4 watch.
And to the left of both of them is the Atalanta pilot, Sarah Sargent.
And your final left is Aaron Rainey, video engineer.
All right. Yeah, join us as we explore this seamount. So this is interesting. Daniel, this is the shallowest seamount that's known in the Palau National Marine Sanctuary.
Yeah, thanks, Blodak, for... so based on what we know, before this expedition started, really 70% of the deep waters around Palau hadn't been mapped, and this feature stood out at us. Now, when we looked at satellite altimetry, so you can create maps of the seafloor using just the sea surface height. So the oceans will bulge over big mountains, and if you measure very accurately the sea surface height, you can deduce what the topography underneath on the seafloor would look like. And yeah, this seemed to be a quite shallow summit, but it hadn't been mapped, and we mapped it for the first time yesterday, and it was quite extraordinary. This feature looks really remarkable. It's about 15 kilometers in width. It has this pretty sharp ridge at the top, and it has several kind of pinnacle-like features on the top. We were hoping to dive this feature last night, but weather conditions unfortunately weren't conducive, and so we spent an extra 10 hours mapping the outskirts of the seamount even more, and then came back in the morning hoping that the weather would cooperate, and it did. So yeah, this is the first glimpses that we're having of this. And it's quite remarkable because, yeah, the top of this is about 500 meters, and then the closest point of land is about 75 nautical miles to our northwest, Maria Island. A little bit to our southwest is Helen Reef and Toby. So we are quite removed here, but this is this one big seamount that sticks out. Now that the Southwest Islands, as I mentioned, were recognized as a ecologically and biologically significant area mostly because of its shallow terrestrial birds and other aggregations of life. And at that time, 10 years ago, there's also about 13 seamounts that were thought that found in this area, had never been explored. And so, yeah, but seamounts are globally recognized as some of the most biodiverse ecosystems in the deep sea. And that's why this area was included in that area. Yeah. Yeah.
All right, thank you for that. The Southwest Islands, there's 2 states here in the Southwest. There's Hatobe State, and we also have Sonsorol State. So Meri is part of the Sonsorol State, and we are very happy to be here to explore what's going on in the bottoms of these oceans.
Yeah. So the Southwest Islands are also considered states of Palau. There are 16 states, and Sonsorol and Tobi are the last 2. And many Palauans in their lifetime actually will never get to go to either of those states. And I was really lucky to actually go to Tobi, which is the farther one, earlier this year. But yeah, so these islands, their shallow water biodiversity is really well known in Palau. And Helen Reef, which is the southernmost island of Palau, I was lucky to visit there, and even we didn't do any diving because I was there for a different purpose, but even just from the beach you can really see things beautiful. There's turtles that come right up to the beach. There's a whole bunch of birds. It doesn't smell very good. But yeah, such a amazing bunch of biodiversity on these islands, so I'm sure it will be reflected in the deep sea portion as well.
Yeah, thanks for sharing all that context. Yeah, truly such a privilege to be able to see these places for the first time and to be able to share it with all of you. And we'll spend the remainder of Lebu's Voyage 2 here down in the Southwest Islands. So we have another 3, possibly 4 dives that we'll have time for. And have several other seamounts, one that is about 120 nautical miles due west of us. The next target.
Yeah.
Okay, push in there.
Metallogorgia?
You're right.
Yeah.
Well done.
What
And
do
then
you
with
call
its
the star on there?
brittle star?
Do you remember its name?
No.
It's called Ophiocreus oedipus.
Ophiocreus oedipus. So Oedipus is like,
Yeah.
yeah. The Oedipus we all know.
Yes, the Oedipus we all know. I'm not exactly sure why it was named that, but
Interesting.
it's probably because of this relationship.
I see. And then Metallogorgia is a black coral?
It's a chrysogorgid coral,
Oh, okay.
so a golden coral. I know this one kind of, the skeleton looks a little more dusky,
Dark,
but
yeah.
usually if you have the skeleton in hand, it looks a bit golden in sheen and color.
Okay.
All right, come on.
Metallic.
Yeah.
Right.
Metallic.
And then this would have 6 polyps? Is it a
8.
hex? Oh, 8.
They're
Oh, it's an octocoral. Okay.
Yeah. Eight tentacles on the polyps.
Got a cookie star?
Could be.
Zoom. Push in there.
It is a cookie star. This is a sea star in the family Goniasteridae. It's very small.
Okay.
Thanks, Jake. And Aaron.
Yeah, another thing about this region here, the Southwest Islands, also recognized for large aggregations of tuna. So that's also or recognized early on as a really important habitat for pelagic species.
Cool.
Oh, what's that fish? It's a fishy fish.
Fishy fish.
Yeah.
Has a fork tail.
It has a fork tail.
Not a whip tail.
Lots of fish. Got 3 of them right there.
Yeah. It's moving kind of like a McTophid. Oh, yeah. And it's gone.
Yeah.
Oh,
and
there's
it's
that.
back. Hey.
It's trying to position itself.
It looks a little bit like Neoscopolis. It's very active.
Yeah, and just looking ahead, so right now, the way the plan is written, we have about 4 hours of bottom time left. So we'll hope to get to the top of this feature. We'll still have quite a bit of climbing to go. And we expect to find some extraordinary things on top of this ridge feature. So we landed on the northeastern flank of the seamount. The seamount has kind of a northwest to southeast orientation. And it is somewhat of a flat plateau at the top, but it has at least 2, maybe 3 very distinct kind of pinnacle features up on top.
So this is a really interesting formation. Is this sediments that's solidified, or is this rocks?
Yeah, I think we've been seeing a lot of solidoclastic sedimentary conglomerates that have been then encrusted in a thick manganese oxide crust. They're not carbonates. We've checked some of the samples we've collected previously, which makes sense given the depths that we have been diving at and collecting rocks at. But certainly it suggests that the ages of these seamounts might be older than we even initially anticipated, given the formation of such sedimentary rocks.
All right, thank you.
Some of those rocks that we collected on previous dives that looked heavy and big and black, like lava rocks, like basalt, we cracked them open and they were just basically semi-consolidated sediment.
Wow.
So I think that's kind of what we're seeing here.
Solidoclastic, it doesn't get easier to say, does it?
No, somehow I always struggle saying it. Solidoclastic.
It's a hard one.
It's a hard one. Yeah.
My favorite geology term is ooze.
Yeah.
Delicious ooze.
Ooze. Silicious ooze and slump. Very technical geological terms.
Yeah, quick, let me zoom on that larger coral.
This is a primnoid coral.
All right, while we zoom there, let's welcome our viewers from Australia, Bermuda, Hong Kong, Mexico, United Kingdom. Shout out to Palau for tuning in. Canada, and we got some from the United States.
Are you
Welcome
pushing there?
all.
So this is a primitive wood coral called Thaurella. There's also a small anthomastus on the rock besides it, and a bunch of ophiuroids in the branches.
That's great. Thanks, Jake.
Okay, come wider.
Yep.
Thank you.
Yeah, so the primnoid corals have been kind of colloquially referred to as the quintessential deepwater octocorals. They are primarily deepwater group. It's about 300 or so described species within this group, but 96% of them are really restricted to depths 100 meters or deeper and to depths over 6,000 meters. Great diversity, genetically really among the most diverse of the octocorals, and still a group that a lot of folks are trying to tease apart how many species, genera, and other groupings are found within this quite diverse group of octocorals.
So is our goal to try to make it to waypoint 5?
Yeah, I mean, that's an ambitious goal, but
It's
I think...
ambitious.
Yeah, I think... Yeah.
I think if we wanted to, we could go straight there.
Yeah.
And that's about 800 meters.
Yeah, I think we should try to go straight. I mean, there was nothing...
Okay.
If we can move in that direction, I don't know if there's been a problem, like the...
Actually, that'd probably be slightly better direction for
Perfect.
the ship, because it's easier when we move forward, and our heading is currently 183.
Yep,
And
I
this
think...
heading would be 220.
Yep, let's go ahead and do that.
All right.
Zooming and zooming.
Two, 2-0.
Yeah, this has already been a quite exciting watch from the little I was watching on the shift ahead of us. But there's still a lot more to come here. The next 3 seamounts that we're planning to survey have all unmapped, and they're all also likely on the shallower end.
I'm not sure what this is. It could be like a flatfish.
A nudibranch.
Push in there.
It looks vaguely fish-like.
What?
It is a flatfish.
Yeah.
Look at it, it's so adorable.
Ah.
Is that its eye?
Yeah, it has both of its eyes are on the side of its head.
So cool.
Yeah.
You
Awesome.
can see its gills moving.
Mm-hmm. It's like, you can't see me. Like, oh, okay.
Look at flat eye.
Totally invisible.
Do we know the name of
I mean,
this
to be
fish?
fair, when we were zoomed out, it was hard to tell if it was a rock or not.
Flatfish?
It's a flatfish.
Okay.
Okay.
Thank you.
Come on.
Oh, it's gonna move.
Oh, I love when they move because you can see the fin rays.
Oh wow,
Like
look at that.
they kind of articulate. They inch around, almost like a polychaete.
Yeah, it looks
Oh, it
almost
does
like
look like
a
that.
scale worm or something like that,
I was
the way it moves.
I was gonna say, now that we zoomed in though, I'm like, obviously it's a fish.
Yeah. But like from here, you wouldn't think it's a fish.
Yeah, it almost looks like a cucumber.
It does.
Yeah, good eye.
I think there may be a fish. No, that's, or is there a sponge?
Oh,
In
the
the
little
still
pink.
cam. Oh, it might be a sponge, a stock sponge.
It's like right in the middle, lower.
Oh.
Oh,
there's
is this the
also-
snail?
There's also something swimming.
Oh, there's the sea cucumber.
Yeah, it's thrusting.
Pushing there.
There's a lot of stuff around here. That's the cool thing about this depth is you start to see more and more stuff. It's a really small anemone, but it's got an interesting mouth in the center of the disc. All right, cool, thanks.
Okay.
Yep.
Come on.
Oh, photo bombing a sea cucumber.
It's trying to get you to watch its workout.
Yeah. Sea cucumber workout videos might be motivating for me, actually. Maybe I would do my crunches.
Just some good music and sea cucumbers crunching.
Mm-hmm.
There's another fish in the still cam. I think it might be the same.
It's an
Oreo.
Oreo.
Oreo. Yeah.
Oreo.
Elsa's got the eagle eye. She is scanning different cameras left and right. That is awesome.
I think her and Sarah might be
Eyes in the sky.
the eagle eyes here.
I'm like containing myself because I've seen quite a few, but they all seem pretty familiar. So
Yeah.
It is all just gonna like erupt when you see an unfamiliar one.
Yeah, sometimes I'll be asking questions about something and I'll realize it's not on screen yet. So I apologize for that.
No, it's great. It gives us more situational awareness.
There are a lot of screens to watch in here. I know some people have counted, but I feel like there are more now than there were.
All right, let's take maybe a quick zoom at that fan.
That might be a plexaurid, one of the parasis.
Pushing there.
I like these shallow dives because there's a lot of stuff I don't know yet. Oh, it's Acanthogorgia.
Yep, that's great.
Okay. Come
Yeah.
on.
You can tell Acanthogorgia because of how fluffy the polyps look. And if disturbed, the polyps can't fully retract. They'll close, but they have a bunch of sclerates along the body that keep them from fully pulling in towards the central axis of the branches.
So are you usually working at deeper than 1,000 meters, Megan?
Usually, well, it depends on what the dives are. So a lot of times when we're doing these ROV dives, we're starting pretty deep. There aren't a lot of seamounts that are this shallow, so it makes for a really interesting time. But my thesis work was actually done all at 450 and 500 meters.
Oh, wow.
So I got to know some of these animals well, but there is a lot of diversity at depths between 500 and 1,000 meters, and it is undersampled. Well, there's a good fish. Let's see that one. It's actually, we can see it from the side, so could get a good ID for it.
Can push in there.
Oh. Who are you? It's got really long fin rays and a really dusky dorsal.
Yeah, and they just looks weird. Are those, I don't know, what's those lines coming out of it?
The really long
Yeah.
fin rays, yeah. So it looks like...
So Jeff says bathygated.
Bathygatus?
Yeah.
Yeah.
Thanks, Jeff.
Yeah, thank you to our scientists in the chat who are watching and helping us with all the IDs. So are we going directly, do we decide to go directly to waypoint 5?
Yeah, we're going to head more directly there. It'll cut out a little bit of the distance, and hopefully we can survey the top of this cone-like feature. We'll get a little bit of the fishiness. Our original plan was to make it to the major summit and survey a little bit there, but we'll have less time in that area in order to see the top of this cone, which I think is going to be really, really interesting.
We love the cones.
We love
a cone.
Yes.
We're going to see another octopus.
Yeah,
Maybe
I mean, our
two.
last cone was like really fortuitous, so.
Fishy.
Oh, yeah.
And it's gone.
I think it's Neoscopolis, but usually they hang out. Oh, look, there's a eel under the rock.
What was that? What was that?
Chilling under the rock.
It just poked out and I got excited. Oh.
Convenient ledge right there.
Yeah, I think it was like, yeah, I'm not dealing with that today. All right. I think it was a conger eel.
There's another one of those faster ones. Up
Yeah,
in
those
front,
fast, bigger fish, I believe, are lanternfish. They're called myctopids.
So Jeff is saying, please, if we see a epigonid in a cave, please try to collect.
Okay, yeah. The epigonids are the deep sea cardinal fishes.
Okay.
They're pretty small, so but they're also pretty dirty. So we'll have to be quick.
What do they look like?
What do you mean, dirty?
Yeah, they dart.
Oh, darty.
Yeah, they dart around really fast.
Very wriggly.
But they're very tiny. They have a double dorsal fin. So they look like your standard split fin fish shape. They're relatively small with a double dorsal fin on top.
Can we also just do a quick zoom on
like
this?
silvery gray in color, and the dorsals tend to be a little bit dusky.
I think it's coral rubble, dead coral rubble.
Yeah, I think so too. Just wanted to confirm because I'm starting to see more and more of this.
Push it there.
Oh, yeah.
Yep, perfect. Thanks, Jake.
Okay. Come on.
Yeah, the epigonus tend to really like areas of vertical relief, sort of like the Oreos. So we don't always see them on these sort of shallower slopey areas, but they do like overhangs and ledges.
Did that just change divisions automatically? I don't mind, but
It's been doing this. I don't know. It's on the fritz.
Huh.
Yeah, it
Weird.
like gets ideas.
Ideas of its own. I
Yeah.
don't know if I like that.
I don't. Now, gee, it's getting spicy.
Their latest software update.
Yeah.
AI.
It didn't used to do that. It didn't do that on the last cruise. This is like a new development. I think maybe it auto scales when the ship gets a little too far off off the page. Oh, a solmissus.
Just wondering what that was.
See that, the jelly,
Yeah.
the plate jelly?
I've been kind of
It was
following
like-
it for
Yeah.
Skinny, and then it goes big.
trying to get it in the blue so I can see
Yes,
it.
please.
Yeah.
I think I can. Oh, dusted it.
I bet the midwater here is really interesting. You usually get a high concentration of really cool midwater animals above seamounts like this.
Yeah, so what Megan was referring there is one of the many reasons why seamounts are really pointed out as being really important habitats of the deep sea. One is just by being these conical features that protrude onto the seafloor, they interfere with the surrounding currents to produce these areas with increased current flow and circular vortices that are trapped on top of seamounts that trap nutrients and therefore create very good habitats for a whole bunch of organisms, many that live on the seafloor. The second one Megan was alluding to, there's a lot of vertical migrating midwater organisms or pelagics that move from 200 to 1,000 meters. When you have shallow seamounts, they intersect with their vertical migration patterns, and so they are basically trapped and concentrated on the top. Another really important feature is that they kind of serve as stepping stones of the deep sea, as basically little islands where the deep water organisms can disperse over large distances of the deep sea. They've also been found to be kind of navigational marks for a lot of highly mobile species that will use these in their large-scale migration patterns. Oh, what do we have there?
Oh, those fast fishes again.
Yeah, one of the myctophids. Maybe it'll cooperate. Jeff can tell us what it is.
Pushing there.
Giving us the whole 360.
It's really adorable.
The eyes.
The eyes are huge.
Yeah.
Better to see you with, my dear. Oh, good job, fish.
He's just doing little circles
I
for
know, I
us.
love it.
The full
Pirouettes.
show.
Pirouettes.
Like,
He has a large scale. I don't know if the scale on the top of the head means anything or it's just a marking.
It's like trying to decide which way to go. I'm not sure.
So epigonus.
Oh, yeah. Okay, you can see the double dorsal
yeah.
now that it's
All right,
cooperating.
so
Beautiful shot.
if we
This
can, we're
is
going
the
to try
one
to
we want to
collect.
slurp.
Yeah, if we are set up to do so, we'll try to collect it if we can, Jake.
We can try.
Yeah. Slurp's already set on.
Okay, I'm gonna come wide. Kind of large with a net.
It is
Yeah.
quite large for this.
It's
slurp.
really big for epigonid.
Oh, and there's something on the bottom too.
Oh, it's a halosaur.
Oh, okay.
Yeah. So maybe it'll just swim right in the hole. Section sampler.
Do you want me to try to slurp it? I don't think that's
Will
gonna
it
work.
fit?
Oh, you're gonna try to scoop it?
I was gonna try the net. I feel
Okay.
like
Yeah,
All right.
I don't think the slurp really has any chance,
Yeah.
to be honest. But
Yeah, it's too fast.
it's too fast. There's not enough jam for a big fish like that. Also, it might be gone now.
Let's just
Swimming
put on a show.
There's
and
another
all
one.
that.
way. It's kind of in
So-
the
Another one
area.
right out in front of
It's
us.
kind of in the area.
There's another one.
They're just taking turns.
And left a little.
Just go back. Maybe we can encourage it to join us.
Hey y'all, I think there's like, Jeff and I think there's a 0 chance you're going to catch this fish with that net.
Yeah.
Oh, yeah.
Okay. Well, I think there's 0 chance with the slurp too, so.
All right, let's move on.
Yeah.
Thanks.
Who was that just...
Sorry,
Oh,
Jeff.
that was Brian in the lounge.
Oh, okay.
Yeah.
But see, like, if it just happened to swim to
Yeah.
the suction sampler.
At least we got some really nice footage.
Yep.
Yeah, you can almost like count the scales. It was being very cooperative. Can we zoom this small fish up here?
Yep. Push in there.
Oh, it's very small. It's a little rat tail. It could be a juvenile. Seems very transparent. Oh, nice. What does Jeff think about this fish?
Nothing yet. But let me.
Oh, there's one of those mega mouth tunicates.
The one right next to it.
Yeah.
Ooh. Will they?
All right.
All right, cool.
That's great, thank you.
So do we know if this seamount was formed by a volcano, May?
Well, everything in this area is volcanically originated, but there's been a lot of geologic processes at work since their formation. So, yeah, all volcanic activity ceased about 20 million years ago, and what we understood is that it began about 60 million years ago in this kind of back arc setting, but a lot of the sedimentary rocks and the thickness of some of these manganese oxide crusts are really puzzling to me and some of the other scientists on board, just because they would take a very long time to form. There are some regions where manganese oxide crust can form faster than the average kind of 5 to 7 millimeters, and we're seeing like 3 centimeters of crust on some samples. So yeah, some of these areas might be a whole lot older than what we thought, but it will be helpful to look at some of the volcanic rocks that we did collect, especially on the previous cruise, as well as some of the ages of the organisms, and dig into the specimens.
Would you like a zoom on this?
Yeah, quick zoom. It looks a little different than...
Okay. The Stauropathes.
Thank you. That was a question from our viewers.
Down,
So
down a few meters? Yeah.
Awesome.
You can
I think Megan is right, yeah.
push in there.
Perfect. Yeah, that's great, and if it's, yeah, the pan maybe up.
Yeah.
Okay.
A little Parantopathes up there.
Just a couple new faces
Mm-hmm.
starting to see.
I have more,
There's another
more
little
zoom
tunfish.
if we need it.
You can push in there.
Yep.
The heterocarpus took off.
That's great.
Okay.
Unless you think this could be like a Cupressopathes.
Chrysopathes?
Cupressopathes.
Come on there.
Yep.
Another one of those flatfish.
Mm-hmm.
yeah, disguising themselves as sea cucumbers.
Big sways.
Yeah, so for those viewers that joined us on previous dives, this type of coral rubble we saw on 2 or 3 previous dives near the tops of seamounts as the predominant kind of substrate type. An indication of that these habitats were sometimes in the distance past more favorable for the reef building corals that created this rubble, but long time has likely since changed, and perhaps they were in a more favorable environment in the past.
Maybe more exposed hard substrate and yeah, a lot of sedimentary processes and erosion going on here.
It's just as my perception, or we have strong currents there from... It says prop wash.
It's this fish.
There's not too much of a current.
The wiggly one.
Roger.
So from Jeff, Cetonurus? I don't know if I'm pronouncing that correct.
Cedonuris.
Cedonuris.
Push in there.
It's definitely getting fishy up here. You sure this is an epigonus too? It's double dorsal. It just has the first dorsal down.
It's like, take a good look.
Yeah.
Are you sure? Collect slurp is what we're hearing from the chat.
These fish are fast.
Yeah.
It's good.
We could just
I mean, we
go
could
on
try.
safari and
Oh, it's right there.
seems to be a lot of them.
Catch them at the right moment when they're doing their spins.
Yeah. I don't know.
Yeah, I have a feeling we're going to see a lot of these.
There's a couple of fish on
It's
the
gonna,
bottom
like, tease
too.
you the whole
Yeah.
time.
Yep.
A lot of fish. Fishes.
Is this one different?
It's a darker color, but I think it's the same.
Oh. They all have that same kind of spin around in a circle.
Yeah, they're
deal.
doing the same behavior.
Yeah, I mean, at some point it doesn't need to be this one, but at some point if we feel like we have a good chance of slurping one, we'll try it, but also don't want to spend too much time trying because these are going to be very tricky.
Maybe this one we could try. Seems to be kind of cooperative. A little bit smaller.
Yeah, let's go ahead if we're set up to do so.
Yep.
Oh, there's something on.
Is that a sea cucumber that's over there?
Ah,
Yeah, I think it's a sea cucumber.
it's making its way in the frame.
Oh no, it's an
Looks like
Oreo.
it's an Oreo, yeah.
All right, turning suction on.
Can you hit PC3? Right next to that one. Oh, never mind, wrong one. Never mind, go back to PC4. I thought that was the lower cam.
Oh.
Now it's startled. Yep.
Yeah, thanks for trying. Yeah, I have a feeling there's going to be no shortage of these guys.
They're everywhere, but they're also very fast.
Yeah.
Maybe we'll get one that's really tired.
Too many circles.
Well, sometimes you'll see them in like really high aggregations. Oh, can we zoom this shiny fish? That one actually looks like an Eoscopolis. It's doing the right posture.
Push in there.
Oh, never mind. Fast.
Fast and startle easy.
Mm-hmm. Well, you know, there's light down here.
Mm-hmm.
They know that they're seen.
Yeah, it's interesting on the first of Lebu's voyage, the last 2 dives of that expedition, so we dove on seamounts, similar depth ranges that are almost directly north of us, 100 to 140 nautical miles north of us. And we did see similar kind of habitat, this coral rubble, but very different in terms of the dominant fauna. There was old type of glass sponge and Gorgonian corals here. The primnoids and the black coral seem to be dominant. Yeah, so I mentioned earlier that the Southwest Islands in this larger biogeographic region, which is thought to be quite distinctive, is also very, very under-surveyed. There was actually one human-occupied vehicle dive done about 30 years ago by a Jamstick submarine. I think about 30 miles west of this seamount, but in a very different habitat that went to depths 6,000 meters and below. And then there was also some recent National Geographic drop cameras directly around the Southwest Islands, but in this enormous area. I mean, it's basically, I really think about half of the sanctuary. They have almost no previous surveys in the deep waters. So really excited that we're getting these first glimpses, and then hopefully another two, 3 more dives to better characterize these environments.
Yeah, this is the Neoscopolis. So shiny.
So shiny.
And we obviously have 0 chance of ever catching this.
Never say never.
Yeah, I shouldn't challenge the ROV pilot. I know better.
Open the box, open the box.
Go over by the Slurp.
It's so pretty.
Yeah, you said it's a
Neoscopolis.
Scopolis.
That's great.
That was beautiful.
Yeah, it's very interesting. It seems to be almost like regular intervals that we see these tiny step-like features.
Mm-hmm. Yeah, we could see that when we were mapping to sort of these terraces almost.
Yeah.
Yeah. I think it's kind of the formation of these sedimentary layers and then the eroding of them away as they become like semi-solidified. But it's very old. Yeah.
Yeah, maybe quick snap zoom on that guy, on that fish.
You can push in there.
So quick. Wait, was that a fish on the ground off to the right? I see it. Just that like gray stripey.
Like a flatfish?
Yeah.
Push in there before the dust.
A bit of rubble, maybe.
Or is it just a weird color? Oh, it's just weird color. All right.
Okay, come on.
It was definitely enticing. It looked like it was something.
Yeah.
Yeah, I'm personally very, very intrigued of what those kind of topographical high points will be on top, because they are kind of 3 of them. They're all about 80 meters
Just a swell.
height and wide. Kind of
Seems
like
to be around 14
dimensions.
decent, but
You would think kind of for bigger coral mounds, but we'll see, hopefully. Megan, what's our approximate range to waypoint 5?
If we go straight there, we are at 615 meters.
Roger that. So still quite a...
It's a haul, but I think we can do it if we're motivated.
Yeah, I mean, I think
on our last watch, we covered,
Yeah.
you know,
We sped up our moves a bit.
we went 1.5 kilometers in 3 hours. So
We were tasked to get to the top, and we did with a few zooms.
Yeah.
Zoom in and zoom in.
Yeah, I mean, I think we should just try to balance that out. Because, yeah, we'll have about 4 hours and some change remaining here. Actually, 3 hours and some change remaining here at bottom time. Should be a short ascent, but...
yeah, well, if we make it to the top of this feature, which is about 550 meters, we get a little more bottom time.
Yeah.
So more land we cover, the more we get to see.
Yeah, that's the bonus.
Jake's down.
Yeah, as always is.
Yeah,
Tell me when to go.
Yeah, I think we should, obviously try to, anything that we haven't yet spotted, try to get a good visual. And balance that out with covering some area over ground.
If we can bump it up to 0.4, our moves, they're not very deep, so there's not like a total amount, like crazy amount of layback or anything, and can stop the ship. And just still do small moves, but do it at 0.4.
Yeah. Okay, we can go a little faster.
If you want to if you want to cover ground, I would kind of recommend that. But,
I think that we should go ahead and do that.
All right. Cool. I will do that on our next move.
Were we going 0.3 before that?
Yeah, we've been going 0.3.
Roger.
Luckily, I have Elsie here, Eagle Eye, that will capture all the things that I'll miss.
Luckily, I put on my glasses today, so.
You're telling us you were doing that without glasses.
Yeah.
Yeah, it's just interesting to me. So we had two dives on seamounts that were very similar in depth and also kind of in the shape of the seamount that were just north of us, and we saw similar habitat, this rubble, but yeah, very different in terms of the dominant taxa. Those were the last two dives of the Libus Voyage 1, where the top was basically the exact same habitat here, but very different in terms of the dominant taxa.
So you're saying it's very similar environment but different species?
Correct, yeah, exactly. So the ground on those areas, the depths, the orientation of those seamounts were different. So this is more of an kind of east-west in terms. Yeah, those were more north-south, but they were quite a bit further north. Those features, whether that's just some break in terms of we've now crossed into a different environment, or it's just
different currents.
Whoever gets there first, that's also been kind of speculated that will dictate what will become the dominant taxa. But yeah, it will be a really interesting data set. Once these 2 expeditions are all said and done, we will have sampled, hopefully, a wide representation of seamounts all around the Palau National Marine Sanctuary, starting in the north, continuing in the east and west, and now in the southern portion, covering several thousands of nautical miles of horizontal distance, and then also wide depth distribution at a depth of 3,200 meters, and this one will be at 500 meters. So really trying to fill in as much as we can of that large knowledge gap that is the Palau National Marine Sanctuary. Yeah, and on our watch, mostly dead glass sponges is all I've seen. I haven't seen a...
All these sponges that we're seeing, we have 2 different ones that I've been seeing. There's a rosellid sponge, Acanthascus, and then there's the farinamatid sponge, Farinamoides. So the big barrel ones that look really hairy are Acanthascus. Oh, you might be able to get this hhmmss in the blue. Yeah.
Push in there.
Nice.
Yeah.
This one's always so pretty to look at.
If we can take the lasers off us, please. Thank you.
So misses.
Mm-hmm.
It's a jellyfish, right?
Yep. All right, we can let this one go.
Come on.
That's a great shot.